Vitamins and enzymes. So these are important things. Enzymes. Anybody want to tell me, like, to you, simply put, what is an enzyme? When you hear that word, what do you think of?
It makes me think of the Krebs cycle because I think of the pyruvate that binds the enzyme.
So enzymes are a major part of pathways, and the Krebs cycle is a pathway. Oh, by the way, Robin, I did get your email. If you could not attend our exam 2 review yesterday, I meant to get that uploaded last night. It didn't happen, so I will get that uploaded as soon as possible today, so you can look over that.
Thank you. I
Yeah.
didn't even remember.
No, you're good. I know that I have to get that done. I'll do it today. Yes, enzymes are a part of pathways, but they don't have to be part of pathways. They can also catalyze individual reactions. Okay, so enzymes allow reactions to happen in a timely fashion for our body. Okay, some of them have to happen very quick, and you're going to see just how quick some enzymes are today. It's probably going to blow your mind. But what kind of biological molecule are enzymes? Are they carbohydrates? Are they lipids? Are they proteins? Are they nucleic acids?
Proteins.
They're proteins, okay? We mentioned that yesterday. So yesterday we hit proteins, which set us up perfectly to talk about enzymes today. And then we're gonna hit vitamins at the end, okay? Vitamins and minerals. So this correlates with chapter 19 in the text. Enzymes, here we go. Are proteins that catalyze biochemical reactions by doing what? Anybody know what we should put in this blank? This actually, you probably learned this in your previous chemistry course, because I know I hit it really hard. I make people draw reaction diagrams. What do enzymes do? They lower something. Anybody remember what they lower? Lowering. You heard of the term activation energy? Was somebody going to say that? Activation energy, okay? And I'm going to abbreviate that. We say EACT, activation energy. Do you remember what activation energy is? Anybody remember what activation energy is? You got a reaction diagram. Remember the reaction pathway, level of energy, the reactants, level of energy, the products. This would be a exergonic reaction. The activation energy is the amount of energy it takes to get the reaction started from here to here. Okay. Anyways, you're not gonna have to draw this, for the exam. But I wanted to remind you what activation energy is. Enzymes lower the amount of energy necessary, so they make it easier, they make it quicker for reactions to happen. Okay. But they're not involved. That's what the rest of our definition says. They're not changed in the reactions. They're not a reactant. Okay. They just bring reactants together, or they take a substrate and cleave it and change it or modify it. Okay, so enzymes have a few parts. I'm going to define these terms for us, okay? Active site. So this is the portion of the enzyme where the substrate binds. So if you're looking at this Pac-Man looking figure right here, my green Pac-Man, we could say that this right here, if I outline it in red, this is the active site, and that our substrate would bind right here, okay? The active site, since this is a protein, the active site is going to be lined with certain amino acids. Now, every enzyme is different. So some enzymes might have really nonpolar active sites. Some enzymes might have really charged, like a lot of positive, negative charges in their active sites. And so they would interact, all enzymes interact differently with their substrates. Okay, so active sites are different. So there's nothing we can classify about an active site, but it is where the substrate binds, and it is where the reaction happens. And it's where the catalytic activity, you might hear people say, occurs. Okay, so it's where the reaction takes place. Okay, this is the place of chemical reactivity for the enzyme. Substrate. The substrate is the reactant. Okay, that is what binds to the active site. There's all different kinds of substrates. Any molecule that is part of a biochemical reaction in our body it could be a substrate for any enzyme. Or it might have a correlating enzyme that it's a substrate for. Okay, but simply put, it is a reactant. It is held in the active site. Okay, there are I'm going to abbreviate intermolecular forces, IMFs, that hold the substrate in place. So if we're looking at our Pac-Man here, what kind of substrates look like they might fit? Well, there's all different kinds. I mean, we could have, and by the way, some enzymes, maybe they just need one substrate. Maybe they bring together two substrates in their active site. So if we're drawing examples like that, if we just have a one substrate reaction, maybe that substrate would look like, just, like a piece of pizza kind of. Okay, just some kind of triangle. Obviously, I'm not drawing this to scale, but some kind of, I'm a little bit of a perfectionist. I'm like, that's not wide enough. Some kind of a triangle substance that would fit into our active site, okay? And obviously, where the red is marked on the enzyme here, this is where the intermolecular forces are going to happen with, if I outline this in green, the substrate here, okay? These pieces come together, and we're going to have different interactions. Hydrophobic, salt bridges, hydrogen bonding, all kinds, depending on what amino acids are there. Okay, but we could have another scenario too, where maybe there's two substrates that are involved. Maybe it's not just one. So if we have two substrates involved, maybe one of them looks like this, a little bit narrower. And then another one could look like, I don't know, like that, okay? And they come together and they both fit in the active site, okay? And again, these are like cartoon pictures that are representing real, actual biological molecules. And they do fit together, okay? Sometimes they fit perfectly. We're going to talk about two different models. One's called a lock and key model, so it might be like a perfect fit, but some may not fit perfectly, but they might fit good enough, okay? So maybe we might have a third kind of a substrate where it's not a perfect fit, but I'll do this one blue. But maybe it's instead of, it's kind of just curved and it's not, as sharp of a fit. Okay. I don't know if I did that justice or not. Okay. And maybe it could become a good fit once it sits into the enzyme. Okay. Just as an example. So we could say this one would not be a perfect fit. But I just want to get used to the idea that there's many ways substrates and active sites can fit together. Okay. Now, I want to define the next term over here, specificity. Okay. Some enzymes only catalyze one reaction. Okay. One, you could say substrate or one reaction. I'll say or substrate. But some enzymes may catalyze like a particular class of reactions or a class of substrates, so they can classify more than one substrate. Okay, so some, if they only catalyze one substrate or one type of reaction or one individual reaction, we'd say that's a highly specific enzyme. If they catalyze many different kinds of reactions or substrates, we'd say that's not a specific enzyme. Low specificity or high specificity, okay? They catalyze just one substrate, highly specific, high specificity. Lots of different substrates may fit, that's low specificity. Okay, turnover number, this is how fast the enzyme is. Okay, so I want you to look at this table. This is what I think might blow your mind. There's a table, it's got different examples of enzymes here. And then it tells you what reaction it catalyzes. I thought this one is such a cool example. So what freaks me out sometimes is to think about how the human body actually probably does contain some sort of cancerous cell like all of us all the time. But some of us grow tumors and some of us don't. All of us have a gene for diabetes. For some of us, it gets turned on, and for some of us, it doesn't. So that like weirds me out thinking about this. We all have harmful substances in our body, okay? One of those harmful substances can be oxidizing agents, okay? H2O2 is a harmful oxidizing agent. And what I love is that catalase here is an enzyme. It's like it just goes around looking to like decompose hydrogen peroxide, okay? Squish hydrogen peroxide, just kill the hydrogen peroxide so that it doesn't harm our bodies. We have enzymes that they're like our like secret agents, okay? They're just protecting us all the time from the inside, getting rid of harmful substances in our body. So this is an example of one. The turnover numbers are widely different. All enzymes catalyze normally somewhere between 10 to 1,000 molecules per second. It's really fast. But look at catalase here. What is 10 to the 7? It catalyzes 10 to the power of 7 molecules of H2O2 each second. One. I mean, that's amazing to me. That's over 1 million, okay? 10 to the 6 is 1 million, okay? Carbonic anhydrase can convert 1 million molecules of CO2 to bicarb per second, okay? Catalase is even faster, all right? So that's turnover number. Other things about enzymes is that sometimes, just like proteins, we said yesterday that we have proteins that are just amino acids, and then we have proteins that are conjugated proteins. Maybe they have like a metal ion or a carb stuck in there to help the protein function in some way, called those conjugated proteins. Same idea with enzymes. Enzymes can work with cofactors, which is a non-protein part of an enzyme, but it is essential to the function of that enzyme, okay, to its catalytic activity. Now, some of them can be like ions and like metallic atoms. Some of them can be just straight organic molecules. If it's a purely organic molecule, then we call that a coenzyme. Okay. Now, why would cofactors be necessary? Why would they be necessary? Why do y'all think a cofactor would be necessary to the function of an enzyme? Well, there's not a real specific answer here. It's just that the cofactor may have some kind of characteristic or property, be it a functional group or some kind of electronic charge that the active site on the enzyme doesn't have. Okay, so cofactors have characteristics. Okay, it could be a charge. It could be a functional group. I'm going to abbreviate. Those are just two examples that the enzyme doesn't have. Okay, or maybe the enzyme has charges on different amino acid side chains, but it doesn't have those charges where it needs it in the active site. So the coenzyme comes in and brings that right to the active site. Now, what is the difference between a cofactor and a coenzyme? Did anybody catch that when I was explaining it? I didn't explicitly say this is the difference, but I did mention.
I might be wrong, but is a cofactor just for organic molecules and not proteins, while cofactors are for proteins specifically?
Not quite clear. Cofactors are more like metallic ions. Okay, so like your main group 1 or 2 metals, alkali earth metals, or transition metals. Those can sit in with an enzyme as a cofactor. Or if it's a molecule, an organic molecule, that's a coenzyme. Okay, so your cofactors are going to be more like metal cations, and coenzymes are going to be organic molecules. All right. So this table actually shows you a whole list of cofactors. And these are different ions. You see they're all different metal ions. And then it gives you the enzyme that it works with. Okay. Trying to see if there's any that I want to point out. You guys know we've talked about alcohol dehydrogenase several times, and we're actually going to talk about it again today. This is the enzyme that metabolizes ethanol. So like if you drink an alcoholic beverage and your blood alcohol content goes up, we have an enzyme that is then responsible for breaking it down. Same idea if we ingest methanol instead of ethanol, which we don't want to do. We have enzymes that break that down, but they have cofactors. Zinc is the cofactor for alcohol dehydrogenase. Okay. All right, this is an example. I want to look at this picture. So this is kind of the, we have computer programs that can give us like the structure of proteins once they're known. So a lot of research goes into even getting an image like this. You have to purify and crystallize your protein, and it's a lot of hard work. But once you get this, we have these beautiful pictures. What do y'all see in this blue structure? This is the enzyme. Aldose reductase is the name of the enzyme. What do y'all see? Do y'all see anything we talked about from yesterday?
Is that a peptide bond?
A peptide bond? Well,
No.
there's tons of peptide bonds. We just can't specifically see them since we can't see specific amino acid residues.
Tertiary or
Yeah, look, what is this? It's an alpha helix, okay? Y'all can see the secondary level of structure, and then you can see the tertiary level of how the alpha helices and everything is folded together, okay? I don't know if you can tell, but like right here, this right here, and that right there, those are beta sheets, okay? So you can see these in the crystal structures of proteins once we get them characterized. Now, that's not the main point I wanted to talk about here. I'm gonna read this gray text. It says NADH gray. So I'm gonna circle NADH. This right here is NADH. Okay, there's another molecule, looks like right up here possibly. Is bound in the active site of aldose reductase. What is aldose? What does that word mean? Y'all know what aldose is now.
it's a carbohydrate with aldehyde on it?
Yes, it's a carbohydrate. It's a sugar molecule, and it has an aldehyde functional group, not a ketone functional group. So there's some kind of sugar molecule bound in here too. And what do you think reductase means? If you had to guess, if you were a betting person, what do you bet reductase means? What is this enzyme probably doing to aldose?
Reducing it?
Thank you. I want you to know when you read the names of enzymes, they seem complicated, but usually the name tells you what it does and who it does it to. Okay, so we're reducing aldose sugar molecules. That's what this enzyme does. So it's reducing sucrose, all right, which is orange. So you see these molecules of sucrose, there might be more than one tucked in here. And we have NADH as the cofactor tucked in that enzyme site to help the enzyme reduce sucrose, okay? Now, going back to cofactors for a second, a lot of them we said are metal ions. When metal ions are bound in the active site, we say the ion is coordinated. So you might hear me say the zinc is coordinated to the active site and it's got a +2 charge. Usually we convey what charge is on the cofactor, but when it's coordinated, we're talking about how it's bonded to the active site. Okay, it's not just floating in there, like it's bound in there. It's interacting with different ions in the active site. So this zinc is coordinated specifically to this active site, whatever enzyme this is. You can see a histamine, a glutamine, and you can see, glutamic acid, glutamate, and then you can see a histidine, another histidine, okay? And these numbers are just telling you where these amino acids sit on the sequence, the primary sequence of the protein, okay? So you can see none of them are right next door. It's not like this is histidine 1, glutamate 2, histamine 3. They're not next door neighbors. They're widely in different spots on the primary structure. Histidine 196, histidine 69, okay? So the zinc is interacting with the nitrogen, the oxygen, and the nitrogen. Why? These are electronegative atoms, right? Nitrogen, oxygen, and nitrogen, they tend to like they have electrons, and those electrons are attracted to positive charge that this zinc has, okay? So we call this a coordinate covalent bond because we're coordinating that metal ion into the active site. Questions on that? This is a table I'm not going to spend a long time. It was just more examples of different coenzymes. So this table up here was different cofactors. These are actual organic molecules. So I definitely, for the exam, I want you to know the difference. If I threw out an example of a molecule and I ask you, is this a coenzyme or a cofactor? I'd want you to be able to classify it. Okay. It tells you what all these different coenzymes do, what they cleave or what they remove from the molecule, and then what kind of dietary, where you can get these. We need these coenzymes. Some of them we have to obtain from our diet because our body doesn't synthesize them. And so this is different dietary molecules that we can get them from. Okay. All right, this is, these tables are really important. So we're about to learn, there's 6 different classes of enzyme, major classes. I mean, there's thousands of different enzymes, but we can group them into 6 major classes based on what they do, okay? So you need to know for the exam, all 6 classes, and then there's like subclasses for each class, okay? And you need to be familiar with the terminology of those subclasses too, because I might use those on the exam. Instead of saying an oxidoreductase, I might just use the word oxidase. And I wouldn't want you to be thrown off by that, okay? So I'm about to walk through this. I'm not gonna read every word of the examples here, but you need to know the 6 main classes. And you need to know, if I were to give you the name of an enzyme, you would need to be able to tell me what kind of class it belongs to, all right? Because the naming goes in 2 parts. One, it tells you the name of the substrate that the enzyme works on. So the example we just looked at on the previous slide, aldose reductase. Aldose is the substrate. Okay, that sugar molecule is the substrate. And that's with every enzyme. It tells you what substrate it catalyzes, and then 2, gives some kind of indication for what kind of class the enzyme belongs to. All right, so this will for sure be on the exam and worth a good chunk of points on the exam. So we have oxidoreductases is the first class of enzymes. Okay, so if you just think about that word, oxidoreductase, it's like we're smushing together the words oxidation and reduction, right? So what do you think these enzymes do? They're responsible for redox reactions in the body. They catalyze redox reactions. They're either oxidizing their substrate or reducing it. Okay, so we have different subclasses. We have just oxidases. They catalyze oxidation by adding oxygen. We said O2 was a very prime example of an oxidizing agent. All right, when we add oxygen to something, we are oxidizing that reactant. Okay, we have reductases. They catalyze the reduction of their substrate. And then we have dehydrogenases. You've heard me say this word before, but this is catalyzing the removal or addition. So we're either giving or taking away hydrogen. If we're adding hydrogen to a compound, we're reducing it, because we said increasing the number of carbon and hydrogen bonds on any molecule is reduction. Removing hydrogen, decreasing the number of carbon hydrogen bonds is oxidation. Okay, so dehydrogenases do both oxidation and reduction, but specifically with hydrogen. Okay, oxidases specifically oxidize with oxygen. Reductases, they reduce. There's many ways they can do that. Okay, the example it gives you here, I mentioned this, but again, alcohol dehydrogenase. You've seen this reaction several times now. I had this on exam 1. Here's ethanol. You have this coenzyme NAD+. This is an organic molecule, and we are oxidizing this. I can draw an arrow. We're oxidizing that carbon right there. We're increasing the number of carbon-oxygen bonds. And the substrate, it has no carbon-oxygen bonds. We're taking hydrogens off, okay? And we're increasing the number of carbon-oxygen bonds. And then it can even go a step further, all the way to carboxylic acid, but here it just stops with an aldehyde, okay? So you recognize functional groups. When alcohols are oxidized, they can be oxidized to aldehydes with mild oxidation, all the way to carboxylic acids with strong oxidation. Okay, so this is a reaction you should be very, very familiar with. So alcohol dehydrogenase is an oxidoreductase, but specifically it's an oxidide. It would be classified as an oxidase. Okay. Another main class is transferase. So this catalyzes the transfer of a functional group. So maybe we're removing an alcohol group from one molecule and we're adding it to another molecule, or maybe we're taking a phosphate group and we're phosphorylating something and we're adding that phosphate functional group. There's all different kinds of ways this can be played out, but essentially we're transferring a functional group between 2 compounds. So there's the subclasses here. I'm gonna zoom in, are transaminases. These are kind of hard words. Transaminases and kinases. So what do transaminases do? They transfer an amino group. What is an amino group? Anybody remember what an amino group is?
Isn't it just a CH bond?
No,
No.
think of amine. Amine is all about nitrogen.
Oh, with the
Amine. NH2. So an amino group is NH2. That's amino. Okay. So the transfer of an NH2 group, or kinases transfer phosphate groups. So you have transaminases, and it's got the word, if you think, this is almost the full word amine in transaminase. So if you think about it, we're transferring amine groups, and then you just add ace on the end. Transferring amine groups, transaminase. Okay, hopefully that word makes sense now. Kinase, I don't have a shortcut for you here, but it catalyzes the transfer of phosphate groups. Okay, so you might see something like this: phosphofructokinase. That is an enzyme that phosphorylates phosphofructose. Okay, fructose essentially is the substrate. All right. Okay. This shows you an example. This is a sugar molecule. It's a ketose. And we're taking ATP and we're adding a phosphate group on this carbon up here. And so it's highlighting it right here. There's the phosphate group. It's in its condensed form. Okay. Sometimes you'll see phosphate drawn out with all the oxygens around it as a phosphate group. But here we're just writing it like this PO3 with a -2 charge. Okay. I'm going to erase that. All right, that's just 2 classes. All right, classes 3 and 4. Hydrolases. These, you'll know what hydrolysis is now because it was on exam 2. Hydrolysis means we're breaking molecules apart and we're using water to do it. That's exactly what a hydrolase enzyme does. Okay, it's an enzyme that uses water. We're going to look at an example later on with chymotrypsin. It uses water to break apart molecules or the substrate in some way. All right, so catalyzes bond breaking with the addition of H and OH to the fragment. We have different kinds, and I love these subclasses make so much sense to me. Okay, lipases, they break fats. Fats are the substrate, and they use water to break apart because fats are named lipids, right? So lipase. Proteases, guess what? They break apart proteins. So the root word there is pro, okay? Proteases break apart proteins with water. Amylases break apart starch, sugars. Nucleases break apart the substrate into nucleic acids. So those names kind of make sense there. The root word usually tells you what the substrate is or what the product is going to be. The example it gives here is with a polypeptide. So we have some kind of protease. The protease is going to crunch this substrate. This is some kind of, it could be a dipeptide or it could be a whole protein, a long string of peptides. But it's going to crunch this bond right here. It's going to break that apart. And it's going to add an H and an OH. Okay, so this shows you the example up here, adding the H and the OH. But here they add hydrogens back onto the amine. If you had the full structure, you would have the hydrogen right here, and then you had NH2. But here it's showing it in its charged forms. Okay, so it's not as clear. But if you get confused, look at this. Look at this right up here. If you have substrate AB plus water, we're adding an OH onto one portion and an H onto the other. So with a peptide bond, I would totally accept if you drew it like this, H and then N2, showing that we added a hydrogen onto here and an OH onto here. Okay, they're just drawing these in their charge form, probably due to whatever pH environment they're in. All right, questions on that? Is that confusing to anybody? Okay. Isomerases. What do y'all think this one does? What word does that remind you of? You're probably reading the definition right now. Okay. This just converts one molecule to an isomer. So it just rearranges the molecule. Remember, isomers are molecules that have the same number and kind of atoms. They're just connected differently. So isomerase enzymes, they don't add anything or take anything away. They just rearrange it and you get an isomer. okay? So they catalyze the rearrangement of atoms in the substrate. Oh, how nice, no subclasses, just isomerase. The example here, you'll see this, and this is part of glycolysis, okay? In fact, I'm gonna highlight, this is a really, because of isomerases, the maximum energy can be obtained from glucose metabolism. So isomerases make that possible. This is a really important step in glycolysis. We're gonna get to glycolysis next week, I think early on next week. So in glycolysis, what's happening here? Let's look, we have a triose phosphate isomerase. So this part down here is staying the same, but look at this middle carbon. It starts out with 2 bonds to oxygen, and then the top carbon is a CH2OH. What's happening is that we are taking probably one of these hydrogens and we're bonding it to this oxygen, it looks like, then we get an OH group, and then we're taking, well, probably it makes sense if we drew it like this. This hydrogen is just jumping ship to that oxygen, and then we get an OH group, and then one of these hydrogens is jumping down to that carbon. So we end up with a hydrogen there. So it's just rearranging the atoms, that's all it's doing. If you look closely, you can usually tell how it's rearranged, all right? Okay, last 2 classes, lyases and ligases. These sound very similar, so they're probably going to be really easy to mix up. So lyases. This is adding or removing a functional group. So remember, transferases, they are transferring a functional group from one molecule to another. This is simply taking a substrate and chopping off a functional group or adding on a functional group to the molecule, okay? Not transferring from one molecule to the next. So catalyze the addition or elimination of a functional group without water. No hydrolysis is involved. Okay, so the examples that it gives, subclasses, you have decarboxylases, catalyze the removal of CO2. You have deaminases, catalyze the removal of NH3. Dehydratases, we're removing water. Okay, think of dehydration when you read that word. Hydratases, we're adding water, we're hydrating. All right. Now I get it, these are all new words, so they're not going to sink in for a little. You're going to have to really read over these, quiz yourself on these, make flashcards if you need to, quiz each other on these, write them out, but practice getting familiar with this language. So for example, what's going on here? We have some kind of alkene. Well, actually we have some kind of dicarboxylic acid. There's a carboxyl group, there's a carboxyl group. But what we're doing is we're adding water and we're adding a functional group. We're adding an alcohol here. Okay, so that would be a hydratase. We'd classify this enzyme as a hydratase because we added water on and added an OH functional group to our molecule. Okay, now the main class is a lyase. So if I said, what kind of main class do hydratases belong to? You would tell me lyase class, okay? Lastly, ligases, they simply bring substrates together, okay? They kind of link up the cars of a train to make the full train, if that makes sense, all right? They're just bonding 2 substrate molecules together. So there you have synthetases, catalyze the formation between 2 substrates. This uses ATP to do that. And then carboxylases, you're adding CO2 to something, okay? You're adding CO2 and a substrate together, which is what you see right here. Pyruvate, this is the first molecule in the Krebs cycle. We're going to get to that tomorrow or Friday, one of the 2 days. So pyruvate is what feeds into the Krebs cycle. And one of the first things that you do is you add CO2, okay? CO2 and ATP, it requires ATP, and you literally just formed a bond. This was the CH3 group, and then you form this new bond between, this was CO2, your CO2 group. And it requires some energy from ATP to do that, okay? All right, I'm not going to read all these big informative paragraphs about these classes, but I encourage you to do that if you are still wondering like what these classes of enzymes do. But let's review. What do we have? We have oxidoreductases, transferases, hydrolases, and isomerases, and then the lipases and the ligases. Sorry, the lyases and the ligases. I can even get those 2 tongue-tied. Okay, so those are the 6 classes. Make sure you know those for the exam. All right, questions on the 6 classes? tomorrow's lab is going to be great practice. It's going to give you a reaction, and just from looking at the reaction, or what you're bringing together, what you're doing, you should be able to tell me what enzyme, what class of enzyme is being used for that. Okay, that's all tomorrow's lab. Practice with that. Okay, how enzymes work. So I'm gonna read you this little abbreviation really quick. You'll see this a lot. E stands for enzyme, S stands for substrate. Okay, so oftentimes in a reaction, there's these intermediate steps, and you'll hear us say the reaction intermediate or the second intermediate. Okay, with glycolysis, there's lots of intermediates when we get to metabolism before you get to your final product. Intermediates look like this. It could be the enzyme substrate, like they've come together, but the substrate hasn't been acted upon yet. They just are bound together. So they were separate, then you bring them together. So that's might be intermediate number 1. And then in the catalytic site, in the active site, all of a sudden the reaction happens. The enzyme suddenly turns it into the product. And there's, I mean, so many different ways that can happen. But this might be, so this would be intermediate number 1. This might be intermediate number 2. And then the enzyme releases the product, okay? So this is kind of the schematic in the shorthand notation for how enzyme mechanisms work, how the different steps of the process. And we call those middle 2 steps the enzyme substrate complex . Substrate complex. And then we call this the enzyme product complex, okay? Same idea. Or you'll hear them referred to as intermediates. But regardless of that schematic, there's 2 different models that fit this, okay? As people have studied enzymes, and this is what I kind of alluded to on our first slide when we were drawing out the different types of substrates, be it 1 substrate or 2 substrates or a substrate that doesn't fit perfectly well into these models. So the first model is lock and key model. So that one's pretty self-explanatory. What that means is that somehow the substrate and the active site fit together perfectly. Just like a door, any lock, okay? Any door that has a lock, that has 1 key that fits that lock. You could have a key that looks almost identical, but if it's not identical, if it's not the right key, it's not going to fit the lock, okay? Same idea with enzymes here. You do have some enzymes that are that highly specific and they only catalyze 1 substrate, and you can see how all the different pieces of the substrate fit perfectly. That one is slightly curved. This one's really pointed. They fit perfectly into the different parts of the active site for the enzyme, all right? We have another model though, and this is how, I think this one's, they're both used, they're both true, but this one is probably what most enzymes would fall into this category, the induced fit model, meaning you have the active site, okay, so this is the enzyme active site, and the substrate here doesn't fit exactly, like it doesn't fit perfectly, all right? But when it gets into the enzyme substrate complex, there's some conformational changes, some shifts in the shape or the change that cause them to then fit perfectly, okay? This is a weird thing I just thought of. Anybody ever had one of those like vacuum sealers? Anybody like freeze? So I think our parents and our grandparents do this. Then they grow gardens, they get vegetables, and then they want to freeze a lot of the vegetables for later use, and they put it in a bag and then they like seal it tight. That's what it makes you think of, is it's sealing it tight, it's tight. There's a tight fit once the enzyme or once the substrate gets into the active site. Conformational changes happen to make that fit really perfect. Then it would catalyze it into the product, and then it releases the products, okay? So that's the induced fit model. So definitely be able to describe the differences to me between the lock and key model and the induced fit model, all right? Draw pictures if you would need to. Now here's a real example with real amino acids and a real substrate, okay? Chymotrypsin. Somebody remind me, this is a great review from yesterday. What does chymotrypsin do? We talked about trypsin and chymotrypsin. These are proteases.
It breaks the bond. I don't remember which bond, but it breaks
On the carboxy terminal side of.
With the aromatic, right?
Thank you, Claire. Yes. Abby and Claire work together on that. Of aromatic amino acids. Okay. Trypsin. Somebody remind me what trypsin does. Where does it cleave? Abby and Claire are not allowed to answer. Somebody else. Trypsin cleaves. Remember? You don't remember? No, you don't. Somebody remembers. On the carboxy terminal side of arginine and, come on, this is painful. Arginine and lysine, okay, positively charged amino acids. Y'all gotta know that. Okay, so here, look, we have an aromatic amino acid, phenylalanine, okay? This is the R group. What does R group mean? That's the side chain, right? So orient yourself to the drawing. Figure out what's what. This is the side chain. This is the alpha carbon, okay? This is probably just the extended polypeptide chain, all right? And this is the carbonyl carbon right there of phenylalanine, all right? So the blue, by the way, this kind of blue shadowed structure, this is the enzyme. This is the active site. The yellow highlighted structure, this is the substrate. Okay, this is the polypeptide substrate. Now in our enzyme, I'm about to sneeze. No, I'm not. Maybe. Okay. In our enzyme, the hydrophobic pocket is what phenylalanine fits into. Why would it have to be a hydrophobic pocket? Because that aromatic acid or aromatic ring there on phenylalanine is really hydrophobic. So it makes sense it's going to bind other hydrophobic, it's going to interact with other hydrophobic interactions or side chains for the enzyme active site. Okay, then later on we have serine and we have histamine and we have aspartic acid. All right, and then you see the functional group of serine here is an OH group. Histidine, you see the imidazole ring with the nitrogens. So we have some polar groups here. We have some electronegative atoms. And what is going to happen? Okay, the enzyme substrate is going to bond together. Okay, we'll get a covalent bond. What happens between, whoops, all I wanted to do was erase this. It's going to happen between this oxygen and this carbon. Okay, so you see that as a green bond right here. I'm just going to, can I highlight it green? Highlighted green. Okay, that bond happens. And you bind, now we have bound the substrate into the enzyme. Okay, then it tells you right here, highlight this in red, this is the cleavage site. So remember, we're cleaving on the carboxy terminal side of aromatic amino acids. So it makes sense that we're cleaving the bond, this is the peptide bond between that carboxy, I guess the carbonyl carbon is what I should say, and then the amine group of the next amino acid, all right? This NH, this is the amine group of the next amino acid, and it's gonna cleave that peptide bond. Okay, so what happens next? Look what happens next. In this figure C right here, now we have broken off. We've broken that red colored bond from figure B. So in figure C, that's broken. Now we have this free peptide. We don't know what it is, but we've broken it off. We've flipped that bond. And by the way, we have to make sure that carbon is following the rules here. In every figure, you notice carbon has 4 bonds. It's just that those bonds are rearranging. Okay, so between A and B, what's the difference between A and B? Look at this carbonyl carbon. Okay, we had 2 bonds to oxygen, 1, 2. Okay, but one of those bonds rearranged, so this bond just kind of fell down in the middle to make the bond between our carbon and oxygen there. But carbon still has 4 bonds. Okay, then this bond is the one that breaks, and those electrons are just going to kind of jump back up to reform the double bond with the carbonyl oxygen. And that free peptide is going to go, but then the enzyme still has to release phenylalanine. Okay, so we have to break this bond between that carbon and the oxygen from the functional group of serine. This oxygen was the OH group of serine, but right now it's coordinated to the carbonyl carbon of phenylalanine. So we've got to break that bond so that we can release phenylalanine once the reaction's over. So that's what you see in figure D here. That bond breaks. We're using water to do that. Y'all see where water comes from? I'm going to highlight it. This and this. So that's why if you read the title of this slide, it says hydrolysis of a peptide bond. Because in the end, we have to use a molecule of water to break that bond between the serine and the phenylalanine and release the free peptide after that once the reaction's been catalyzed and it's done. Okay, there's a lot of moving pieces. I am not going to make you draw out a reaction mechanism, but I could very well show you an enzyme active site with different like amino acids, and I could ask you, what kind of reaction do you think this would catalyze? Okay, what kind of functional groups might fit here? What kind of functional groups would this substrate need to have to bind here? Okay. So I wanted to walk you through this as an example to show you how enzymes do work. They're very specific. It's very tedious, but this is as deep as we're going to get into any kind of mechanism. There's all kinds of things here. I briefly want to run through these 4 effects. Okay, obviously things have to come into proximity. Okay, so there's this what they call the proximity effect. Okay, you've got to bring things close together for the reaction to happen. Things have to bind into the active site. Things have to bind the right way, the orientation effect, okay? That's what enzymes do a good job of. They not just bring molecules together, but they orient them the perfect way so that the reaction happens at the lowest possible energy, all right? And exactly the position necessary for reaction. The catalytic effect. All the right functional groups are right there in the place of the active site of the enzyme so that things can happen quickly and they can be catalyzed and done, all right? So it provides all the basic acidic, other types of interactions needed. And this is provided how? Through the side chains of amino acids. It's the side chains of amino acids that find themselves or are folded perfectly into the active site that provide all those functional groups necessary for the catalysis. We call that the catalytic effect. The energy effect. Because things are already brought into perfect position and perfect orientation, the energy is lower. We don't need as much energy to work to find each other and to orient ourselves if I'm just the substrate looking for my other substrate to collide and to react and clear the energy barrier. Okay, that's why we say enzymes lower the activation energy. Okay, it lowers energy barrier by inducing strains in the bonds in the substrate. Okay, don't necessarily worry about that part so much. All right, now, so we talked about different kinds of enzymes, the active side of enzymes. We've defined things like substrate and turnover, but what affects enzymes in the body? There's several things that affects enzymes. Sometimes enzymes can function at really, really high speed. Sometimes they're impaired and they don't function properly. There's all different kinds of things that can speed enzymes up and slow them down, okay? So what kinds of things would do that? Well, number one, the concentration of substrate. This would be the main thing that affects how quickly an enzyme works. If there is a lot of substrate, then there's a lot of work for the enzyme to do, and it's going to act quick. If there's very little substrate, then it's not going to function as quickly, okay? Not all the enzyme active sites will be filled to capacity. So I want you to, the other thing I might as well go ahead and fill this is temperature. Okay, so I'm going to discuss these 2 graphs for just a minute. But before I do that, I want you to consider the fact that your body has a finite amount of resources in it, right? You're only so big. So we have a certain number of enzymes in us, a certain number of proteins. Okay, so consider the fact that the concentration of enzyme in your body is not changing. Okay, we don't just add new enzymes. Now, sure, enzymes can be recycled, and our body can generate new enzymes as it makes new cells. But that's kind of like the number of enzymes dying off in our body is probably equal to the number of enzymes. It's creating the amount that it needs. It's not going to create more than that. All right. So you have a certain number or certain concentration of enzyme in your body. So that's not changing. Okay. But the substrate, that can change based on what we eat, if we're taking any medications, if we're doing physical activity, what kind of environments we're in. The substrate levels change in our bodies constantly. Okay, so the enzyme doesn't just need to be turned on to full power all the time. It just needs to respond to the amount of substrate in our body. So think about this. What's going to affect enzyme activity? We might call this the reaction rate, the rate that the enzyme's functioning. Think about how substrate concentration correlates to this. So if we have X amount of enzyme in our body, as the substrate concentration increases, then the rate of the reaction would increase correspondingly. Okay, so if we double the concentration of substrate, the rate of the enzymatic reaction will double as well. And that's what this line is showing you here. Okay, as it's kind of pretty vertical, it's increasing. The rate is increasing as substrate concentration increases at first. Okay. But at some point, since we have a finite amount of enzymes in our body, finite concentration, at some point we will have added enough substrate that all the enzyme active sites for that particular substrate are filled. And at that point in time, we say the enzyme is saturated. What does that word saturated mean? Anybody heard that word before? I often introduce it when I'm talking about solubility. Anybody ever tried to dissolve like sugar into hot tea or like salt into water? It'll dissolve at first. You have a cup full of water, hot water or hot tea or whatever. It'll dissolve a certain amount of sugar, a certain amount of salt, but at some point you've hit the solubility limit, and we say the solution becomes saturated. That's where I introduced it to my chemistry students prior. So that may have been the term you heard it in. But that word saturated can also apply to biological molecules here, enzymes. Okay, if we have X amount of enzymes, a certain concentration of enzymes, we have X amount of active sites. And when every single one of those active sites is filled to capacity, we say the enzyme's saturated. It's like this: if you go to a theme park, okay, and everybody has their own favorite ride at the theme park. Mine is not the Superman ride that goes straight up and down. I hate that ride, okay? But there's a certain number of seats on every ride, okay? And that's why things get backed up and lined up, because they're, at peak season, like in the summer, there could be tons of people, customers or whatever, in a theme park. And so each ride becomes saturated very quickly because all the seats are filled. Okay, that's the same idea here that we're talking about with enzymes and active sites. When every active site is filled with the substrate, we say the enzyme's saturated. Okay, at that point, we hit a maximum in the rate, and the enzyme just can't go any faster. Okay, no matter how many people line up at a roller coaster, the roller coaster takes a certain amount of time, right? It's not going to go any faster just because there's more people. Same idea with the enzyme. Okay, we've hit a maximum rate. So at that point, what do you guys think would happen if we just raise the substrate concentration? Do you think the rate's going to continue to increase? If you're looking at this graph, what does it show you? If we just keep adding substrate, like keep extending the arrow this way, will the line continue to go up? Will the rate always continue to increase? You can just subtly go like this or like this. Watch it. No, right?
No.
It's gonna level off, and we're gonna hit a maximum rate, no matter how much substrate's present in the body. Okay. Okay, so that's concentration of substrate, how it can affect our enzyme. Temperature can affect the enzyme to a certain degree. If you increase temperature, you're gonna increase the movement of molecules. They'll move faster and faster and faster. So at first, that can really increase the rate of the reaction if you increase temperature up to a certain point. But then if you keep increasing the temperature after that, things are going to start to backfire. Okay, because temperature, what did we say yesterday at the very end of lecture? Heat does what to proteins at a certain point?
Denatures it.
Denatures them, and enzymes are proteins. So if we keep heating things up beyond 50 degrees especially, you have no enzymatic reactivity, no enzymatic reaction, okay? So temperature is helpful only to a certain point. And then lastly, pH. So most enzymes, they have an optimum activity around physiological pH, 7.4. And then they can function at pHs slightly below that or slightly above that, okay? In the stomach, though, the stomach is an exception. We have an enzyme pepsin, pepsinogen. That might be the zymogen, but regardless, it can function at like a pH of 2, okay? Because our stomach has a lot of acid in it. So there's some enzymes that are an exception to that, but most enzymes function between 5 and 9, and above or below that, they're just not going to have their optimum activity. They're going to begin to unravel or denature. All right, I think this question kind of hits on what I just asked, actually. What do we mean when they say the enzyme is saturated? I literally just explained that. So I'm going to skip over this question. When the enzyme is saturated with the substrate, how does adding more substrate or more enzyme affect the rate of the reaction? That's a great question. That's things for sure I would take time writing out your answer. That's a great way to prepare for the exam. I definitely want you to know how concentration of substrate and enzyme correlate for the exam. Okay, next question. Enzymatic activity is shown for 3 different enzymes as a function of pH. What does that mean as a function of pH? How does the rate of the reaction, how does the rate of the enzyme, the speed of the enzyme change with changing pH? Okay, so as a function of pH in the following graph, what is the optimum pH for pepsin, curve A, urease, curve B, and alanine dehydrogenase, curve C? Okay, so all you have to do, if I give you some sort of graph like this, is you're looking for what is their optimum, at what pH is their optimum activity. So this is the y-axis is enzyme activity. So obviously highest activity should correlate to a certain pH. So for pepsin, what does it look like for you guys?
pH 2.
pH 2, okay, pH 2 for pepsin. What do we say B was? Urease. Okay, so pH 6 for that one. What do y'all think for C? C is less clear for the alanine dehydrogenase.
pH 9.
Anybody agree or disagree?
pH 9.5.
There we go. Right around pH 9. But specific, I want you to be as specific as possible. 9.5 actually would be a more accurate answer there. Okay. What time is it? 9:41.
No, it's 10:05.
Oh gosh, do y'all see that on my iPad? Up in the
Yeah,
corner?
it's stuck like that. It's
9:41?
never moved.
It's like
I've
every
looked
Yeah,
at
I've
it
noticed
like every day.
It's
It's
not
always
working on the
9:41.
iPad.
Does that screw y'all up so bad some days?
oh yeah.
Oh my, why does it do this?
It's like every time you're on campus, it does that same exact date and time.
Seriously?
Mm-hmm.
January 9th, 9:41. All right, well, don't look at my iPad. Thank you, Robin. Let's take like a 2-minute break. I need to go grab some water. I'm gonna come right back because we have enzyme regulation. Inhibition is its whole little unit here. I'm gonna go grab some water, and after that, I think we just have vitamins and minerals left. Okay, so hang tight. You can turn your screen off if you need to. Don't leave the Zoom, but I'm low on water. I'm gonna grab some. Be right back. Okay. I'm back. All right. Okay, I think we will jump back in. Y'all there? How many slides do we
Yeah.
have left? Let me look. Well, it says 12 out of 22, so 10. Okay. All right, so far we've defined what enzymes are, the different pieces, different parts. We've started to understand how substrates might react in the active site. We looked at the different classes of enzymes and then how substrate and temperature and pH, substrate concentration, temperature and pH affect enzymatic activity. Okay. Inhibition. So I think the two hardest parts of this lecture are going to be getting down the 6 classes and then being able to identify if you're given a reaction or if a reaction is described to you, identifying what class of enzyme that is. I think the other hardest part of the lecture is what we're about to talk about, inhibition. There's different modes and different kinds of inhibition. Now, defining them is probably not going to be the hard part, but actually applying them to like real examples, that's kind of hard sometimes. So we have a practice problem just after we talk through inhibition that is going to be a really good practice problem, okay? So enzyme regulation. Why is this important? I want you to appreciate this for a second. Think about your body every single day, every time you eat a meal, every time you breathe, every time you get up to go do physical activity or move in general, every time you digest a medication, every time you go to the bathroom, like our enzymes are constantly regulating everything our body does, everything you do, okay? So this is fantastic, but we don't just need enzymes on at full blast all the time. So our body has this really complex, amazing way of ramping things up and ramping things down. I mean, think about just keeping your blood glucose levels constant, okay? That is a feat that's phenomenal in and of itself, okay, that our bodies do that. When we experience so many changing environments internally and externally every day. So regulation is everything. It's everything about how our body operates. But with that, we have activation and inhibition. This is how regulation works. So we want to turn enzymes on. We want to activate them sometimes. So any process that increases enzyme activity, we call that activation. Okay, ramps it up, turns it on, increases it. Obviously, inhibition is the opposite. Any process that decreases enzyme activity, all right, is inhibition. So we're going to focus on inhibition for just a minute, all right? How do we go about inhibiting enzymes? There's different ways we can do it. So we have these different modes. Number one, reversible. It's called reversible uncompetitive inhibition. And you're going to see why it's called that in just a second, okay? So in this type of inhibition, the inhibitor is another kind of molecule, and it does not compete with the active site. Okay, so it maybe binds somewhere else or just next to the active site. It doesn't have to, this inhibiting molecule that inhibits the enzyme does not have to look like the substrate. It can look completely different. It can be similar to the substrate, but doesn't have to be. All right, so it's uncompetitive. It just, the picture here, I think, does a really good job. Okay, so this enzyme, the substrate is the orange part. The inhibitor binds not in the active site, but you can see how it would prevent the substrate from coming back out or even going in if it had not gone in yet. All right, so it's going to inhibit. That's a good example of how an uncompetitive reversible means this inhibitor can come off and on. So when we no longer need to inhibit the enzyme, the competitor can come off and then the enzyme can function normally. All right. So it binds the enzyme substrate complex so that the reaction occurs less effectively, or it can shut it down altogether. It might just slow it down, or it can completely stop it. And this is a really, I like these little bullet points too. Overall, the maximum rate of enzyme efficiency is lowered. All right. So if you have a high concentration of, actually, sorry, that's on my next one, competitive. This one, it doesn't matter how much substrate you have. You could ramp up the concentration of substrate. It doesn't matter because they're not competing. Okay, it's uncompetitive. Now, when you have reversible competitive inhibition, the inhibitor actually is, it's another biological molecule that does look similar to your substrate. Okay, and it binds in the active site or a portion of the active site. So if we have some kind of substrate in orange, a competitive inhibitor might just have a feature that fits part of, actually I'm going to do this in a different color. It might just have a feature that fits part of the active site, but altogether it stops the substrate from coming in and reacting with the enzyme. Okay, so think about this. If I had 1 million molecules of my substrate, but I only had 100 molecules of my inhibitor, which one's probably going to win most of the time? The substrate is, right? So these are completely concentration dependent. If you ramp up your concentration of substrate, the enzyme will keep functioning properly because it will compete out the spot for the inhibitor in place of the inhibitor. Okay, so that's why if we read below, did I read all this? Inhibitors similar to the substrate in size or functional groups binds the active site, preventing the substrate from binding. Okay, this is what I was getting to. Overall maximum rate of enzyme efficiency can still be reached. You could still have that enzyme functioning powerfully at its maximum rate, but only if you have high enough concentration of substrate, okay? So it could compete out the inhibitor, right? For uncompetitive inhibition, it doesn't matter how much substrate you have. So examples of this, glucose 6-phosphate and methanol poison. I wanted to briefly mention this. So glucose 6-phosphate, this is part of glycolysis. The very, very, very first thing that happens to glucose when it comes into the cell is we phosphorylate it, okay? What kind of enzyme does that? Anybody remember? What kind of enzyme throws a phosphorylate group or phosphate group on there? Kinase, okay? So it comes glucose 6-phosphate. We phosphorylate it on carbon number 6. All right, so we have
You
glucose
said what
6-phosphate.
enzyme?
Sorry, Cameron?
You said what enzyme?
A kinase. That's up on your table earlier. I was just seeing if anybody remembered really quick.
Okay.
You're good. Kinases. So we phosphorylate glucose at carbon number 6. So that's why we call it glucose 6-phosphate, or you'll see me write it G6P, probably a lot in the future. In glucose 6-phosphate, when there's enough of it, like when we have plenty of energy in our body, because ultimately glucose is metabolized to make ATP, right? So if we have enough energy in our body, we don't need to just keep metabolizing glucose through the pathway of glycolysis to make energy. We have enough. We can use glucose for other things, or we can store it as glycogen. So glucose 6-phosphate, so if we just have plain old glucose, step number 1 in glycolysis turns it to glucose 6-phosphate, and we use a kinase to do this. This becomes an inhibitor, and it'll come back around. If the enzyme is our kinase right here, it'll come back around, and the product here inhibits the enzyme. And our body's brilliant by doing this, so that way we don't make a whole bunch of stuff we don't need, okay? So it's a competitive inhibitor. It binds in the active site. It competes out glucose when we have made enough energy. We have enough glucose 6-phosphate. We can use glucose to do other things, all right? Methanol poisoning. Methanol in the body is immediately metabolized to formaldehyde. Okay, what kind of molecule is formaldehyde? What kind of functional group do you think it has? It's half the word.
Aldehyde.
Aldehyde, right? So let's draw it. Anybody remember what formic acid is? I had this on your exam. It was an ester though, so it's formate. Formic acid is simply this. This is what formaldehyde is. Okay, you got the aldehyde group right here, but we just have a hydrogen on the other side. This is formaldehyde. This is really toxic to our body. And so we don't want methanol to be metabolized to formaldehyde. So what happens is that ethanol will come in as a competitive inhibitor, and it will block the enzyme that, so we have some sort of enzyme that catalyzes the metabolism of methanol to formaldehyde. And ethanol looks a whole lot like methanol. It's just different. Instead of a meth group, we have an ethyl group. Comes in, this is the competitive inhibitor. I'm just going to abbreviate CI. It's the competitive inhibitor, and it will come and it will shut off that enzyme so that we don't make any formaldehyde. And that methanol is then just excreted from the body. It's brilliant, okay? So we have all kinds of different examples for the way these modes and models of inhibition work. Questions on that? Okay, these two are the same. I just threw in this third one down here. So we have irreversible inhibition, and it's exactly as it sounds. We just have some kind of molecule that comes in and it binds the active site permanently, and it literally just shuts off the enzyme. Okay, so this only happens once in the enzyme's life, because after that, we can't turn it back on. But it's irreversible. No matter how much substrate, you could add all the substrate in the world, but it's shut off the enzyme permanently So this can happen with different heavy metals, okay? This is why we obviously don't want to ingest heavy metals. So anybody heard about like lead-based paints and how those used to be so dangerous? People used to paint their houses with lead-based paints in the 50s and the 60s, and we've like outlawed those. We have new kinds of paints now because paint would chip and little kids would eat paint chips, Lord have mercy. And that's very dangerous, okay? Or the fumes in general. So that's one example. Heavy metal ions can bind in the active site and just shut the enzyme off. Mercury, what should we watch out for in our diets, particularly concerning mercury? How could we ingest mercury?
Through fish.
Specifically tuna, okay? So tuna, and it's the tuna fish, they acquire mercury just from the ocean. And then we eat tuna and we acquire mercury from eating tuna. So those are just a couple of different examples there. All right, allosteric. So those are the different modes of competitive, non-competitive, irreversible inhibition. Now we have a totally different kind of inhibition, okay? It's called allosteric control, feedback inhibition. All right, allosteric control, what is that? That is the binding of a molecule at another place on the protein that's not the active site, completely different spot on the protein, all right? Causing a conformational change, a shape change in the protein that affects the active site, and it affects how the substrate binds, okay? We have two kinds of allosteric control. We have positive and negative. All right, so a positive allosteric regulator, it opens up a previously unavailable active site. So the example here is actually on the right, positive allosteric control. So this is, the green is our allosteric regulator right here, okay? And there is seemingly no active site, okay? But the arrow is pointing to an unavailable active site. There is gonna become an active site available right here. That's where the active site would normally sit. So once this positive regulator binds right here, now the active site is made available and the substrate can enter. So that means it's kind of like an extra molecule that just turns the enzyme on. Makes a conformational change that makes the substrate or the active site available for the substrate. The opposite of that is negative. The substrate and the active site are running and gunning. Available, but then this allosteric, a negative regulator, comes in and binds a different spot, not the active site, and it shuts the enzyme off. It causes a conformational shape change that affects the active site. Now the substrate can't bind. Now these pictures are obviously just cartoon pictures. It doesn't mean that the active site completely disappears, right? Or it doesn't mean that the active site was completely gone and then it completely appears. It could be a very minor change, but the substrate, the point that these figures do a good job of is showing you the substrate can now enter, it can no longer enter, okay, once that change happens. Questions on that? That was pretty straightforward. The regulators do not have to look like the substrate because they're binding a completely different spot. So they're not competing with the substrate. Okay, this is an example of feedback inhibition or feedback control. And this honestly kind of sounds a little like that glucose 6-phosphate example I gave with our kinase in the first step of glycolysis earlier. But basically in feedback control, this occurs in pathways where the product of one reaction is the reactant for the next. So that is a pathway. So look here, we have a linear pathway. It's not branched. In the body, we hardly ever have linear pathways. Things are often branched because the body's so interconnected. But here we have reactant A, and enzyme 1 turns reactant A into reactant B. Okay, or you could say reactant B is the first product, but then product B becomes the reactant for step number 2. So B is the product of enzyme 1, but it's the reactant for enzyme 2. And enzyme 2 converts it to C. C is the product for the second step, but then it's the reactant for the next step. This is how a pathway works, okay? And in feedback control, all right, each individual step, by the way, catalyzed by its own enzyme. In feedback control, D here is gonna come back and it's gonna bind to enzyme 1 and it's gonna shut enzyme 1 off, okay? So for example, I'm gonna go back to glycolysis because it's just such a great example, and this does happen. In glycolysis, what I showed you, this would be like glucose, would be A, and then the G6P, glucose 6-phosphate, would be B. So the example I gave you earlier, it is a kind of feedback. It was a competitive inhibition that we talked about because the product for step 1 was inhibiting the enzyme. So here, this is a little bit different because it's not the very next product. It's the end product of the whole reaction that comes back and it binds that first enzyme. And why would it do that? Why would it not just shut off enzyme 2 or 3? Anybody have a thought on why our product D here would not just shut off the enzyme right before it? Why does it have to go all the way back and
Does it have
Fine,
something
enzyme.
to do with them like making too much and then stopping halfway? Does that make
making
sense?
too much, stopping
Like-
halfway? Yeah, we don't want the body to waste resources.
So
We
you
don't
wanna
want to
just-
make things we don't need, right? So here it looks like in this pathway, B and C are intermediates that are only used for the production of whatever D is, okay? And if we don't need any more D because the body has plenty of it, we don't need B and C, right? So it wouldn't make sense for this pathway to run all the way to C and then for D to just cut off enzyme 3 and stop it there, because then we don't need B and C for anything. All B and C is used for is to make D. So the body doesn't want to waste resources. So in feedback inhibition, good grief. There we go. D is going to cut off the pathway at the very first enzyme, okay? So that we don't waste time and resources. Okay, energy is a resource, making B and C when we don't need them. Does that make sense? So that is feedback control. Where feedback is what? I mean, think about just the word feedback. For example, you go to an interview, you get the job or you don't get the job. You probably want some kind of feedback on how you did in your interview, like what you did good at, what you did bad at. We receive feedback all the time, okay? Some of us are great with receiving feedback. Some of us don't like to receive feedback. But here the body has to give feedback, okay? It has to tell enzymes and pathways when we have enough of any particular product or reactant for the next thing, okay? So that's why we call it feedback control. All right, lastly, another form here of enzyme regulation. These are our last two: covalent modification and genetic control. These are our last two. So let's start with covalent modification first. Some enzymes are synthesized in non-active forms known as zymogens. Zymogens, zymogens. By the way, I'm looking at the time. I know we only have like 3 minutes left, but what I'll probably do is finish. Let's get to this practice problem and do this practice problem. Then we'll take a break. We'll come back and we'll finish with vitamins and antioxidants and minerals. Okay, then we'll do the lab. So even if I go past 10:30 here, I just want to get to this practice problem. We're 2 slides away. Okay, zymogens. Okay, so these are non-active forms. Think of it like this: like an enzyme is already made and created, ready to go. It's just asleep. Okay, so that's like a zymogen. So we have to do something to wake it up. All right. So if somebody came to you while you're sleeping and cut off your right toe, that's a terrible example. That'd probably wake you up though, right? But that's what happens here. We're literally going to clip something, a part of the protein or the enzyme off to get the awake form and the active form of the enzyme. So the removal of terminal polypeptides or some kind of chemical modification is going to yield the active enzyme in this case. So this is exactly how we get chymotrypsin. So I want you to look at this. If this is the sequence, okay, now I know it looks like it's all laid out in a linear form, the pink bar here with these little blue spots on it. But if this is the sequence, so this would be like the primary structure of, we call it in the sleep form, chymotrypsinogen. So you'll often see that for zymogens. Notice the name is OGEN, and you'll see that that OGEN ending. Pepsinogen, trypsinogen. Okay, that's the zymogen form. Okay. And everything that's pink is necessary. These are amino acids, okay, a representation of amino acids that are needed in the active form, all right? Now, we're not seeing what amino acids are there. It's showing you some disulfide bonds. So we got some cysteine residue somewhere in there, but this is just the sequence. Now, in the sleep form, in the zymogen form, we have these blue portions that are not needed, okay? These are residues that are intentionally put as part of the primary sequence to keep chymotrypsin asleep, okay, or inactive, because we don't always need it. But when we do need it, we need to have a quick way to turn it on. So we have other enzymes. Trypsin, in this case, is going to come and cleave chymotrypsinogen, and it's going to turn it on. It's going to take out the parts that keep it inactive. And when those parts are removed, now we have chymotrypsin, which is the active form of the enzyme, and it's ready to go, okay? Now, again, you're gonna think, well, it's in 3 parts now, but remember, think of it in its folded structure, okay? It's not just in 3 parts, it's folded together. You have all these intermolecular forces holding the protein together, and we've just removed small pieces here and there, but the protein's gonna hold together, okay? You're looking at the primary sequence because I think this is the easiest way to explain the difference between a zymogen and an active form of the enzyme. But in reality, things are already folded up together. They're not laid out straight, and then we have to fold them together. Blank is another kind of covalent modification in which, phosphorylation, reversible. Okay, reversible phosphorylation is another kind of covalent modification. So oftentimes too, instead of clipping things off the sequence or off the enzyme, we can phosphorylate it. We can put a phosphate group on there, and now we have the active enzyme, all right? And where do we add phosphate groups? We add them specifically to amino acids that have alcohol functional groups on the side chain, okay? OH, serine, tyrosine, and threonine, they all have OH groups on the side chain. And so that is where we can phosphorylate, right? You definitely, I'm going to highlight this. This is important. Okay, if I gave you a sequence and said, you know, what points of the sequence could you phosphorylate this enzyme to activate it? I wouldn't want you to tell me just any residue, okay? So again, kinases are the enzymes that phosphorylate things. So it'd be some kind of kinase would phosphorylate, or you can dephosphorylate. Kinases can remove phosphate groups to activate or deactivate. Okay, but we have specific name. Oh, sorry, phosphatases are the ones that do that. This is a picture of phosphorylation and dephosphorylation. So let's say we have some enzyme called phosphorylase B, and then the active form is called phosphorylase A. So if we have a serine residue, this is the sequence here, this little blue bar, and we have these OH groups pointing off at some point in the sequence, all right? This is a very, very simplified picture, but letting you know you got some sort of serine residues or tyrosine residues at some point, and you can use ATP, and the kinase here will take ATP and phosphorylate that group. So it would look like this afterwards. Now that oxygen from the OH group is directly bonded to the phosphate group. So we can phosphorylate it, and now the enzyme is more active or turned on. We can remove those. The enzymes that do that are called phosphatases, not kinases. So kinases and phosphatases can regulate phosphorylation to ramp up enzyme activity or inhibit enzyme activity. Any questions there? I know I'm not talking about like why water and everything. This is just used to remove the phosphate group. Okay, we got to add hydrogen back onto this oxygen. And we add, so water, we're adding hydrogen back on to the oxygen there, and we're adding an OH group back onto the phosphate group. Okay. So that's water is split apart and used. It's kind of to hydrolyze that reaction. Okay, so that is covalent modification. Last one, genetic control. And I literally just have this sad little sentence about genetic control. But basically, we don't need all enzymes at all stages of life. Okay, think about a baby. Babies grow, okay, their first several years, I mean, up through adolescence, even into our early adult years, our bodies are growing. So we have certain enzymes, they're called growth factors, okay, that regulate how our bodies grow. But we don't need those when we're 60 and 70 and 80, right? We don't need to keep growing. So those enzymes are needed early in life, but then we shut them off or we down-regulate them for later in life, okay? Think about puberty. Right? When you hit your teenage years, a lot of stuff starts changing. Your hormone levels start changing. Enzymes regulate all of that, but we only need those things for a certain season. Okay. And then we're matured and we no longer really need those enzymes. So because of that, we have to regulate certain enzymes genetically, which means so it's the regulation of enzyme activity by control of just even producing the enzymes. So we make enzymes early in life or later in life or a certain stage of life, and then when we no longer need them, we stop making them. Okay, so that is genetic control. Some enzymes are only synthesized early or later in life. So 2 examples, or I just really will give you one example, but growth factors. Fibroblast growth factors. Okay, these would be used early in life. Okay, another example, I will give you another example. Alcohol dehydrogenase. Okay, we said infants are especially susceptible to alcohol or ethanol content in the body because they don't have a lot of this alcohol dehydrogenase. It's down-regulated. Enzymes shouldn't need to metabolize ethanol because they shouldn't be exposed to it, right? But later in life, we do upregulate alcohol dehydrogenase so that we can metabolize substances like ethanol should they be put into our bodies. Okay. This is a summary, and then I'm going to do this practice problem, and then we're going to take a break. So I'm going to keep both visible because you're going to want to look at this summary. I think these questions are pretty challenging because we've just covered a lot of content and there's no way it has sunk in yet. So I'm going to be here to help. But let's talk for a minute about scenarios A, B, C, and D one at a time. What type of enzyme regulation would be best for the following situations? A, an enzyme that becomes overactive during a disease, okay? You catch a cold, you have some sort of viral infection. You need your immune system to go into action. Your immune system is regulated by enzymes. We want those enzymes to be working at full speed, right? To combat any sort of disease in our body. We just don't want them overactive. We want them active. We want them reacting and working at full capacity. We just don't want them overactive. So what kind of inhibition do you think we could use to just slow the enzyme down just a little bit?
Maybe feedback control?
Feedback control. So in feedback control, we said it's the product of a certain pathway cycles back to inhibit an early stage enzyme or an earlier step in the pathway via allosteric control. So if this enzyme is working on a particular disease molecule, once that disease molecule is broken down or cleaved or killed in some form, would it make sense that those products would come back and feedback regulate that enzyme to shut it off? Does that make the most sense? we obviously, I don't think we want, genetic control doesn't apply here. Okay, because you can get diseases at any state and time of your life, right? It can happen to infants, it can happen to old people. So it's not that we want this enzyme to only be present at certain stages of life. So I'm going to say not genetic control. Covalent modification. Covalent modification doesn't necessarily apply in this particular state. Maybe we don't want the enzyme on if the disease isn't present, but that's not the specific scenario we're looking at. The disease is present in our bodies. We want the enzyme on. We just want to slow it down a little bit. Okay, so it's not that we want to have in a sleep form and an awake form. That probably is okay for a different scenario, meaning if we're sick or we're not sick. But in this scenario, we are sick. We just want to slow our enzyme down so it's not overactive. So could you think of a type of inhibition that would still allow the enzyme to function at full capacity, but just slow it down a little bit?
Would it be reversible competitive inhibition?
So let's explore that. If we have a reversible competitive, remember when we were talking about competitive inhibition, we have some other kind of modulator or regulator molecule that could come bind in the active site or near the active site if it's uncompetitive and slow the enzyme down, okay? And the amount of disease in our body, if it's competitive, let me go back to competitive real quick. I want to point one thing out. The overall maximum rate of the enzyme efficiency can still be reached, but only at elevated concentration of substrate level. So here our substrate would be our disease molecule, right? We want our enzyme fully saturated and breaking down any disease molecules in our body. And if we had some sort of competitive inhibitor that could, I mean, this is the picture we had for that, that could bind in the active site and block the enzyme if we needed it to. So here the driver for combating this disease in our body would be the amount of disease molecule. So does that sound like a good fit? Like the enzyme still functioning at maximum capacity depending on how much of the disease molecules in our body? Again, I'm telling you, I think this problem is a challenging one. That is for sure the best fit. A competitive, inhibition. Okay, and it's reversible. So essentially, the regulator that could slow the enzyme down and the disease molecule, the concentration of those are going to determine the enzyme function. It's not going to shut it off completely, but the enzyme is still going to run at full speed.
You said competitive, not uncompetitive?
Competitive, reversible competitive inhibition, Because in non-competitive inhibition, it wouldn't matter how much of our disease molecule you have. The concentration there is not going to influence enzyme activity. But in competitive inhibition, the concentration of your substrate, here the disease molecule, would completely regulate enzyme activity still, which you want to happen. You just want to slow it down a little bit. So if you have something competing for that site a little bit, it would slow the enzyme down some. Does that make sense, Cameron? I hope so. What do y'all think about B? An enzyme needed only when there is low blood glucose. An enzyme needed only when there's low blood glucose. So think about when your blood glucose levels drop.
So is that uncompetitive?
Let's look at this. Hold on. I'm not ignoring your comment, Cameron. I just want to think about that. We have an enzyme. I'm going to draw a little enzyme here. and it acts on glucose. Okay, when blood glucose is low, think about insulin. Okay, this could be insulin. When blood glucose is low, we need to chop up glycogen, which is our storage molecule, and release some glucose into the blood. Okay, but when we have enough glucose in the blood, we want to stop releasing glucose into the blood. We don't want to release too much. Okay, so the concentration of glucose, the enzyme needs to be reading that somehow, or that needs to be sending information back to the enzyme somehow. The concentration of glucose in the blood needs to be sending information back. So if we have a lot, I'm just going to write G for glucose. Okay, if we have a lot of glucose, all these glucose molecules in the blood, we don't want the enzyme making any more glucose. If we have very little glucose in the blood, we want the enzyme releasing more glucose. So somehow this molecule needs to come back and communicate to the enzyme, hey, we've got enough, stop doing what you're doing. What does that sound most like to you? Does it sound like competitive? Is there any other molecule that the enzyme glucose would be competing with here? That doesn't quite fit, okay? The only thing the enzyme needs to be reading is the levels of glucose.
Would this one be feedback control?
Robin, what'd you say?
Would this one be feedback control?
Yes, it would be, okay? The level of glucose is like, if you think about this chain up here, it's like glucose is D. And when we have, there might be multiple steps, we don't know, to get glucose into the blood. But when we've got enough, we want to shut off that reaction because we don't need it anymore, okay? And we don't want to waste resources in the process. So absolutely, feedback is a great way. That would be the best way for the body to handle this situation. Okay, what do y'all think about C, an enzyme that springs into action when a traumatic injury occurs? Okay, so we're not always dealing with traumatic injuries, physical injuries to our body, but at a moment's notice, that can happen, okay? My son is learning to walk. He was at my parents' the other day, and he fell and gashed his forehead, all right? So he starts bleeding, okay? So obviously, the one thing your body wants to do when you start bleeding is stop the bleeding. So you need your blood clotting process. A great example here would be blood clotting process. We need that to jump into action immediately. So what we need is for those enzymes to be ready to go, but we don't want them active. We don't want to clot our blood all the time. So we want them in an inactive form, and then in a moment's notice, we want one little quick change and we want them active.
So
What does that sound like?
Is it zymogens?
Yes, that is what would fit best here. Does that make sense? Why it would be zymogens and not something else? If it were genetic control, then it might take a long time for our blood clotting process to respond. If we have to first go make our enzymes and fold them and get them in the body where they need to be. No, we want them in the body ready where they need to be. We just want them asleep. And then when the injury happens, we want to wake them up and go. Does that make sense? Okay, what do y'all think about D? An enzyme needed only during adolescence. So at a certain stage in life, we need this enzyme. Once we get past adolescence, or even before adolescence, we don't want it. We don't even want it around.
Genetic control?
Absolutely, Maggie. Okay, now let me put you at ease. I'm not going to have 10 of these questions on the exam, but I think this is a really good way to start. You are going to have to apply inhibition to actual scenarios. So I probably will have something similar to this. But I think these are hard, okay? Because all of this is new and there's many, many different ways this can play out in the body. So I think there should be some good practice problems on this stuff with the recommended textbook problems. I forget, I need to go look at our homework assignment to see what we do have on there as well. But we've been going for a long time. That practice problem took a while. So let's take a break. Let's meet back 10:50. Let's meet back at 11:15. Okay, 11:15, that'll give us an hour and a half to finish up and then get over to our lab assignment for the day, okay? If there's any questions, I will stick around, but y'all take a break. I'll see you back on at 11:15.